Welcome to Neurally. Let's pick our brains. This video is part of a series about how our five senses work. Today we'll be covering the sense of hearing, or audition. So, how do we hear? First, we need to understand the properties of sound. Anything that can move air molecules can generate sounds, since sounds are simply variations in air pressure. Specifically, areas of compressed air, known as high pressure, or rarefied air, known as low pressure, form sound waves. The frequency of the sound wave is the number of compressed or rarefied patches of air that pass by our ears, and this frequency is measured in hertz, the number of cycles between air patches per second. Frequency also determines pitch, so higher frequencies have higher pitches and lower frequencies have lower pitches. It's also important to note that there are some sounds which our ears cannot hear. The other important property of sound is its intensity, which is the sound wave's amplitude. Intensity determines the loudness we perceive, so louder sounds have higher intensities. Now we must understand the structure of the ear. The ear is split into three parts: the outer ear, the middle ear, and the inner ear. The pinna is on the outside of the ear and it collects sound. The entrance to the inside of the ear is the ear canal, which ends at the tympanic membrane, also known as the eardrum. Sound waves vibrate the eardrum, which is connected to a sequence of bones called ossicles. These bones include the hammer, anvil, and stirrup, and they transfer movements of the eardrum to the oval window. The last ossicle pushes the oval window, which causes the fluid in the cochlea to move. This movement of fluid creates ripples in the basilar membrane, which bends the hair cells on its surface. The hair cell movement triggers impulses in nerve cells, sending neural messages through the auditory nerve to the brain's auditory cortex. The most important part of this process occurs when the vibrations from sound waves are converted to neural signals. The process involves the movement of hair cells on the basilar membrane, which is located in the cochlea. The cochlea is the fluid-filled coiled tube in the inner ear, and if we uncoil the cochlea, it's evident that the basilar membrane vibrates differently across the length of the cochlea, making it sensitive to different frequencies of vibration. The basilar membrane is wider at the apex than at the base, so at the base, which is more narrow, we hear higher frequencies, and at the apex, which is more wide, we hear lower frequencies. Hair cells each have about 100 stereocilia, and when the basilar membrane moves, the stereocilia are bent, which opens potassium channels and depolarizes the cell. This depolarization signal is sent to the auditory nerve fiber, and after passing through the cochlear nuclei in the brainstem, the signal reaches an area in the thalamus called the medial geniculate nucleus, also known as the MGN. The signal passes from the MGN to the auditory cortex, which processes auditory information. It's also important to understand how the sound's intensity and frequency are encoded. A sound's intensity, which contributes to loudness, is coded by the firing rates of neurons and the number of active neurons. Yet a sound's frequency is a more complicated story. A lot of frequency sensitivity is the result of the basilar membrane, but more importantly, in the brainstem's cochlear nuclei, there is a map of the basilar membrane. So cells next to each other often encode more similar frequencies than cells far away from each other. This organization is called tonotopy, and it allows for auditory information to be precisely transmitted to the auditory cortex. Now that we know how our brain receives sound, we must understand how our brain interprets a sound's location. Interaural time difference is the time it takes for a sound to reach one ear versus the time it takes to reach the other ear. An area in our brainstem called the medial superior olive encodes the horizontal difference by keeping track of when the sounds come from the left and the right ear. As for vertical differences, the pinna's convolutions delay the direct path of the sound wave, so our brain knows how to analyze these differences. When our brain processes auditory information, we hear the world around us. But our brain processes more than just sounds. The brain helps us perceive other kinds of information, like smells, which we will learn more about next week.